Hi everyone. So I'm a faculty member at the University of Chicago. I develop statistical methods, and more recently I've been kind of trying to learn deep learning in genomics and try to apply them to our data. So when you have to give a title, I don't know, at the time I said it, I will try to have at least one slide that is related to this, but yeah, you will see. So we will be talking about predicting the epigenome. Do I have? So does it bother if I have this on the side? No? Yeah, I like having that. Okay, so since this is a biobank, I don't know where to go. I stay here. Okay, I just stay here so I can see you and the slides and my laptop and all that. Okay, you probably all know what a genome-wide association study is. We have been running these studies for the last 20 years. We discover more and more loci, but most of them we don't know how they act on the phenotype. The general consensus is that most of these variants, since they are in non-coding regions, they act through regulation of gene expression. So this is just an example from the 2015 paper from Albert et al., where you see, let's say this is the GWAS locus, and perhaps this blue allele, this person here has the blue allele that allows the transcription factor to bind more easily. That leads to more transcription of the downstream genes that you see more of them here, and that translates into more proteins. And if the gene is deleterious, it will lead to higher risk of the disease, whereas this other person that has the yellow allele doesn't allow the transcription factor to bind as easily. Fewer genes get transcribed, and then fewer get translated into proteins. That leads to lower risks of the disease. So this is the general, consensus on how, one of the main sort of drivers of how GWAS loci are acting to alter phenotype. So based on this idea, back more than 10 years ago now, I proposed this method that we call PredictScan, and now it's more kind of widely known as TWAS method, right? There are multiple different TWAS methods. At the core, they are all doing the same thing. So let me go through this. I know you know what a GWAS is, but I kind of, to focus your attention in this terminology, so when I show you this later, you know what I'm talking about. So what is a GWAS? Is this, in a GWAS study, you collect individuals. So these are the rows on these matrices where you have the genotype matrix, the phenotype matrix. So you measure a phenotype, could be height, blood pressure, disease status. So that's your phenotype. You will have one value for each individual in your study. And your genotype matrix will be, so rows are people, columns are different genetic variants. I call them SNPs to keep it simple for just terminology. So a GWAS is, the simplest thing you could do with this data, which is a linear or maybe logistic regression of Y, regress on each one of these columns one at a time. That gives you this SNP level results table that we call GWAS summary results, and these tend to be very widely available. You go to the GWAS catalog, you can download for these tables for many of your favorite phenotypes. So since we know that we don't know, we don't even know what are the causal genes, right, from this GWAS, all these GWAS loci that we are discovering. So one way to go about would be, why not run a transcriptome-wide association study where you run, instead of running associations against against SNPs, you do against gene expression levels. Right? You measure gene expression levels, do exactly the same thing, and run this regression that will give you gene level results. At least your results will be genes and not, RS number 128 something. So the problem with that approach is that, measuring gene expression is a lot more expensive than measuring than genotyping individuals. And also, there are many tissues that are not accessible. You can't just take people and get a piece of their brain and sequence their RNA. And the other thing is, if you find differential expression between cases and control, that doesn't necessarily mean that that differential expression of the gene caused the disease, but it could be the other way around, right? Reverse causality. So because once you have diabetes, for example, your transcriptome will be all messed up, and then there will be the expression will be different, not because that's causing the disease, but because as a consequence of the disease. So as a way to address those problems, we propose this method where you take, so these things on top is what we already have from GWAS, right? The field has spent, I don't know how much money, maybe billions of dollars in doing this over and over in many, many different people. So we have this data. So the proposal was to take the genotype data and predict gene expression levels. So instead of measuring gene expression, predict, and that tends to be in general cheaper, and then you do the same thing and you will have gene level results. So in order to be able to do that, we need to be able to predict gene expression from genotype data. So first question is, can we do that, right? So, GTEx consortium, probably you all heard about, is another very large effort that was started in order to answer the question, what is how the genetic variation affects gene expression and how that is affecting the phenotype of interest or your complex disease. So in GTEx, you have same idea, genotype. So this is about 1,000, maximum 1,000 individuals. These are organ donors that they consented their organs to be extracted, and in addition to their organs, like about 54 different sites of their bodies were extracted at the time of organ collection, and they were genotyped and then RNA-seq. So it was a huge effort that ended roughly in 2020, and now there's another one that's developmental GTEx that is going on as well. So using this data, you can predict gene expression. So here rows are people, as always, and columns here are measured gene expression levels from RNA-seq data. So we feed the model each one of these columns, gene expression in, let's say, blood or brain or something like that, against not just one at a time, but all the SNPs at the same time, although we use cis-SNPs, so SNPs that are nearby the transcription start of the gene of interest. So we fit this linear model. Here X, we will have X is the genotype data. So we'll have multiple, it could be 1, 10, hundreds of SNPs that ends up predicting expression levels of this gene. So we save these weights into a database that we call PredictDB. And this is so every time you want to predict gene expression, you can go to this website, download the weights, and then use your genotype data to predict gene expression. Okay, so how well do we predict gene expression? And here is just 4 cherry-picked examples, ARAB2, NAT2, PEG6, and ARAB1. And you can see on the x-axis is predicted expression. And please ask questions. And if you don't ask questions, I may ask you the questions. So, be warned. And on the y-axis is the observed expression. So you can see there's a pretty good correlation, R-squares of 80%. We're not used to these kinds of predictive performance usually in genomics, but for these genes, we do pretty well. So if you look at all the genes that were, for example, expressed in whole blood in this example, you can see that, so each dot here is a gene, and the black curve shows the heritability of the genes. So heritability of the gene, by that I mean what proportion of the variation in the gene expression is due to the genotype. So you can see that to the right, we have genes that are more heritable or genes that have expression that are more heritable. Yes?
I do have a question about the prediction data. So
yeah.
Let's say you had a variant that was like a potent hypertension risk factor. So the people who have that risk factor, they're much more likely to take antihypertensive medication.
Yes.
And that will alter gene expression.
Yes, that's why.
The pathway will be
the other way.
Gene to hypertension to drug to altering the pathway. So is there any effort to try to regress that out or understand that relationship?
Okay, yeah. Thank you for that question, because that's exactly the reverse causality issue. And that's why we don't do observed expression, differential expression. What we do is genetically predicted expression. Let me go back to here. So maybe here. So what we do is we predict expression based on your genotype data. Your genotype is germline. It doesn't change because you have the disease. So that's one of the advantages of using this approach instead of observed expression.
Right, but these are all still taken from people, right? So what if the cadavers that were getting sampled, all these tissues?
Oh, I see. Yeah. So EQTLs, right? It's the relationship between genotype and gene expression because of different contexts.
Yeah.
We have not seen a huge difference. Like many studies have collected, for example, in mental health disorders or sorts of things. I mean, there are some papers that show that you kind of, when you use more relevant patients or samples, you may get more EQTLs, more relationships that are more relevant. But in general, we don't see a lot of difference between cases and controls. But that is something that has been investigated.
I see.
So, yeah, there was a question there.
My question will also be very similar. In the base, we are basing on the association between the genotype and the phenotype. And the phenotype, let's say, this would be the same example, hypertension. Hypertensive people, non-hypertensive people from the three health records or their health, they will be getting hypertensive medicine. How would one differentiate between the hypertensive medicines versus the hypertension's actual effect? Technically, like just based on this, on a very statistical equation, you would need to know the hypertension medicine's effect on a normal person and unmedicated hypertension person to actually separate the effect between disease and the drug given for the disease.
So we are not doing drugs here, right?
But
the effect, are we talking about the effect of genotype to expression?
Yes. Is it the genotype?
Yeah. So if you're worried that the gene expression will be altered, so if you want to investigate the sort of normal regulation, you could just use the non-hypertensive people, right? Then you wouldn't have and then you can compare, are those EQTLs, right, those relationships different from the people who do have EQTLs? And when you combine them, do you get things that are consistent? And in general, that's what we have seen.
Okay. But how do I understand the hypertensive medicine's expression versus the hypertensive people?
So you would have to do a different study for that, right? So you would have to collect hypertensive people that are, were on that medicine and do the study. But these are, in general, population-based studies where there may be a few hypertensive, or maybe more, because these are people who have older people in general. But yeah, and you can separate those, use them as covariates, and then do some sensitivity analysis. But definitely it's an important question to ask. Yeah. Mm-hmm.
Just a comment, the fact that these variants also mitigates these indirect effects, because it's more likely to be direct.
Yeah, good point. Yeah.
Maybe you can get there, but how do you estimate the weight of like
Yeah. It's a simple elastic net type of regression. There are other ways. People have tried BSLLM, Lasso. In general, elastic net for gene expression prediction, I haven't seen it outperformed by other methods. But more to come with traditional methods, let's say. Okay, yeah, thank you very much. This was, I was worried that people would say, oh, you're always talking about the same thing. But yeah, I'm glad that we cleared this up. So, okay, so many advantages to doing gene level associations versus SNP level association. Let me skip that. So how much time do I have left? Okay, and so let me skip all this. So this method is currently being used as post-GWAS analysis. There are other post-GWAS analysis I recommend people to run, and you want to have multiple lines of evidence to decide whether a gene is worthy of following up, developing a drug for a certain disease. But it has many advantages, but of course, like any method, many limitations. So it's obviously not very good at predicting genes whose heritability are low, right? If it's not heritable, then you cannot use genotype to predict gene expression. But those are more likely to be disease relevant, right? So that's a problem. There was a paper from Hakamanesh, from Jonathan Pritchard lab talking about that, the effect of selection in the kinds of genes that we can predict well or find EQTLs for, and so on. But let me not get okay. Let me continue. Yeah. So also other things. Prediction don't transfer well across ancestries. Effects of rare variants, we cannot, they're missed because they're rare. We may even not find a variation at all. You need relatively large sample sizes, hundreds to thousands. And also there's another kind of general consensus that we are coming to, that steady state expression, or what people are calling steady state expression, which is like bulk expression data you find, may not have the regulatory process that are relevant for disease. Maybe disease develops during development or after some perturbation, and those are things that get missed when we just look at this so-called steady state expression data. Okay, so then we ask the question, can we leverage deep learning methods to build our next generation of PredictScan or TWAS method? So, probably you know what this is, but let me go over it for those few who don't. So what is deep learning? It's a machine learning method that uses neural network with multiple layers. It's deep because you have many layers. You have the input layer, that's the input data, your X, and then the output layer, your Y, the thing you want to predict, and in the middle you have a bunch of hidden layers. So a deep learning model is simply a mathematical function that maps input data, xy, x1 to xn, to output data or your prediction, your y. And the parameters of these functions are the weights that connect the nodes. So that's all, you need. I mean, that's actually pretty simple if you think about it. So the deep learning model that we looked at for the purpose of predicting gene expression that will help us find causal genes for diseases is Informer. How many of you have heard of Informer? A few people, right? AlphaFold? Anyone? Yeah. Nobel Prize last year. Alpha Genome? A few, yeah. So Informer is the predecessor to, you know what I'm trying to say, Alpha Genome. So this is a deep learning model. It has 240 million parameters. So, if you're a statistician, you say this cannot work. But, magically they do work. So that's, anyway. So this model, a deep learning model, what it is just the function, right? You input your data, you have your output data. So the input data is DNA sequence. So you take a DNA sequence of a person or the reference genome, for example, and you would take about 300,000 base pairs. It depends, so it varies, but let's say 200,000 base pairs. You can put it into this deep learning model that will do all these calculations through using 240 million parameters. And as output, it gives you this matrix that is 896 rows by 5,313. So each one of these columns corresponds to a different epigenetic tracks that may be DNA-seq data, ataxic, so chromatin accessibility, histone modification in different cell types, tissues, many cancer types they have used, transcription factor binding from ChIP-seq experiments. They have 2,000 of those. Cage experiments give you sort of expression at the transcription start sites of genes. And the rows, the 896 bins are, of course, each bin is 128 base pairs. So in total, it ends up being covering a window of 120,000 base pairs. So you plug in, let's say, 120,000 base pair sequence, you put it through this model, and it gives you predicted epigenetic tracks. And what does it look like? So these are a few of the tracks, right? The track will be the whole genome. I mean, if you run it by chunks, and then you will have the whole, you can run it for the whole genome, but you will have to do it by these chunks because you cannot get more than 120,000 base pairs out of this model. So here in green is the predicted tracks. So these would be the reads in your experiments. And then in blue is the other way. Green is the experiment, blue is the predicted by informer. What do you think? It's very good, right? Yeah. So the new version, the successor to informer, some people call it Informer 2.0, is Alpha Genome. This is from DeepMind. So DeepMind decided to use their brand name for this model. So it allows 1 megabase long input window. And then it gives you, in addition to all those things I said, RNA-seq data, cage was already an informer, DNA's attack were already histone modifications, transcription factor binding, and a bunch of splice sites. And the thing engineering-wise, it seems that it is a feast. And they have a resolution of 1 base pair instead of 128 base pairs as before. But this model is, the new, style from Google is not to share their models. They said they were going to share it, but we'll see. Apparently, it's under review. So the first author said that when it gets accepted, they will share, I think. Anyways, so since it's not public, we are still not using it, although we have tried a bit. So they have an API, a notebook. So it's a Colab notebook. I would suggest if you're interested, go to their, search, Google it. It's Alpha Genome Docs Colab. And it is pretty nice. You can just plug in, you just make up a sequence and it will give you the epigenetic feature predicted for that. You can also give it a location in the genome and it will predict for the epigenome for that. Anyways, so that's, we will be working on that when the model becomes available, but in the interim, we're still using Informer. There's another one called Borzoi that from the same last author is, we haven't seen it to outperform Informer for our purposes, so we are still using Informer. Okay, so yeah, this is something that, that's a difference that is worth sort of spending a little bit of time. So the way Informer is trained, they use the reference genome. So there is no population, rare variation, nothing. There is no LD, it has no idea what you're talking about. All it knows is sequences of DNA, of bases. So, so it learns that certain patterns means that there's going to be, they look like transcription start site or a promoter or an enhancer and so on. It doesn't have those labels explicitly, but it kind of behaves like that. So, but the thing is, it learns across the genome, right? So you predict the epigenetic track and you compare the epigenetic tracks. Essentially, if you think of them as gene expression tracks, it tells you whether gene A is more highly expressed or lowly expressed than gene B. Right? So that's what this method is doing, and that's how it is being trained. So it uses the genomic dimension. But when we do GWAS or TWAS or things like that, what we are interested in is whether a gene A is more highly expressed in cases versus controls, right? We want to know the orthogonal dimension, which is, so we want to, so essentially we want to look at the orthogonal dimension. That's all I want to say. So, in principle, this thing that has learned across the genome, what do you think? Can it extrapolate and give you variation across individuals, which it has never seen variation across individuals, right? So we tested that, and other people have tested, and these things have been published. So if you take a person's genotype data, and then you take the reference genome and you mutate the, the places where the person has the mutation, the alternative variant, the alternative allele, and then you plug it into Informer, it gives you the whole matrix this whole matrix. And then one of the columns is expression level in LCL, in lymphoblastoid cell line. And you can sum the things that is around the transcription start site of that gene, and then you can sort of quantify expression of the predicted expression of a gene for, this is one gene, I don't have the name here, and the dots here are different people. So you can see that on the x-axis is observed expression, y-axis is predicted expression, right?
Is this from GTEx?
This is Giovadis, which is from, this is 1,000 genomes people. They have, LCLs, lymphoblastic cell lines. There has been RNA sequencing done. In 2013, there's a newer version now that may be better quality, but yeah. So it's not GTEx, but similar to GTEx. So you can see it's pretty good correlation, right? It's one of the 4 genes I had shown you before. And then another gene, we get this. So what do you think? Yeah. So then what people have said is that, and then there was these papers, these 2 papers back to back came out at the end of 2023, saying, these methods seem cool, but then at the personalized level, they are useless because they give you.
Were they evaluating all stuff at the population level in their initial paper when they evaluated this?
The Informer paper.
Yeah.
Informer, the genomics people don't look at variation across individuals usually, right?
Yeah.
No, they only, so human genome is the reference, human reference. HG, I don't know which one they used, right? So no, they didn't look at the variation across individuals. Okay.
Can you go back one slide, please? Yeah, hi, I'm James. I'm a postdoc here at UCLA.
Uh-huh.
I'm curious, all these genes that you're kind of going through, are you, I mean, obviously you're doing bulk RNA sequencing and trying to predict with bulk, and so I'm just kind of curious, like, what tissues are you looking at?
So,
and are you also just, like, the ones that are correlated, is that just a function of just, maybe these are just housekeeping genes and aren't, like, cell-specific or something along the lines of that? I'm just kind of curious. Like,
yeah, so maybe this is one tissue, LCL lymphoblastic cell line, in this example, right? The observed is lymphoblastic cell line. The predicted is one of the columns in Informer, so it was very easy for us to test that. So I don't think I will have time, but to go through that. But the application of this is going to be to train predictors at the cell type level using single cell data.
Yeah, I just wonder if there's like an extra dimension that you could like integrate into maybe one of those plots that you had before, where you have like heritability, but then you also have this extra dimension per se of like, what would be a good way, like relative expression that you have, because maybe the reason that you're not like getting these strong correlations sometimes is because of like you're like missing like a factor in there, like a dimensionality that I think that would be really helpful for when you're plotting. I don't know that I can probably explain maybe later.
Yeah, we can talk more, but this is a pretty strong correlation. It's not, the thing is, it's in the opposite direction.
Yeah, but it's an inverse correlation.
Yeah,
it's
so.
the complete opposite of what you're looking for. So that's why I'm trying to figure out. Like,
yes, it's yeah. So a lot of people have been trying to figure this out and trying to train the model in a way that doesn't have this problem. And as far as I know, nobody has been able to solve it. But yes, it's an open problem. Yeah. So based on that, people decided that this is not good. You cannot use it at the population level. But the thing is, so in one of the papers they show this is the different methods, Informers, and some competing methods here. So the correlation across genes, right? So they tend to be around 0.6 R. If you do the correlation in the genomic dimension, but if you do the correlation in the across individuals and you average across genes, it's mostly 0, right? Around 0. Whereas PredictScan, our method, gives but obviously, PredictScan cannot give you this, right? It's 0 here. Anyways, the point is, if you look at plot the prediction performance of Informer against the prediction performance of PredictScan, which in PredictScan or TWAS methods, the goal, sort of the loss function is the correlation, right? So it gets the correlation right, PredictScan. That's what this thing here is saying. But looking at this and say, oh, it's 0 on average, is kind of ignoring a lot of actual data information that you have, right? So you can interpret this and be a pessimist and say, you can drink water, but eventually you'll die, you will do nothing. Or you have the water, it may not be perfect, you drink it. So we drink it and we use this tool that I think has tons of information about the regulatory effect of DNA, and we build a sort of single cell PredictScan. So I think I'm run out of time, but so yeah, essentially what do I say? We train the predictors, we do a bunch of things, and I can talk more with you later. I was supposed to talk about this part, and the title, right? The title of the talk was using a single sample size of 1, right? And we do that because Informer is using just the reference genome, right? And then when we do the training with single cell data, we don't need many samples. Like if you have more samples, you average them, then you will have less noise. So, but we are leveraging this variation and not the variation across individuals, which we lack with single cell data because they are expensive, you don't have enough sample sizes, or if you have the sample sizes, you don't have enough reads, right? So yeah, we use the model, and then the final, after doing a bunch of things that we solved many problems, we show that, for example, in this case, we ran TWAS using our new Informer-based predictors in beta cell here. You compare to the traditional approach, because you can do the traditional approach with the current data, and we find many more genes compared to the traditional methods. And then finding many more genes doesn't necessarily mean that it's better. And we compare to a set of expert curated likely causal genes, and for those genes we also find our results are more significantly correlated. So this seems promising. We apply to other diseases with similar results. So yeah.
Never mind. I'll talk to you later.
Okay, take home message. We use Informer, which is a sequence to expression models, as a foundation model. You probably heard about this foundation model because it costs a lot to train this model, and then you can use cheaper approaches to fine-tune them. So that's what we did. We created CTPred, which can train predictors of single cell type specific expression using very few samples. And we can improve our power to annotate genes to GWAS loss size, and use them for disease drug targets. So that's all. Thank you. I'm happy to answer more questions and show you more.